I'm Nick Willett. I'm a postdoc in Bob Goldberg's lab, and I'm going to present our speaker today. Our speaker is Dr. Mike Davis. Mike got his bachelor's from Rutgers University, or Rutgers College, sorry.
Same thing.
He went on to get his Ph.D. at Emory University and then did a postdoc at Brigham's and Women's Hospital at Harvard Med School. He opened his lab back in 2006 at Emory University, and he is an assistant professor in biomedical engineering here and at Emory in the School of Medicine. His lab researches the delivery of small molecule inhibitors and antioxidants for cardiac regeneration and stem cell therapy. And with that, I will present you Mike Davis.
Thank you, Nick. So Nick had a good opportunity to get back at me because I heckled him during his thesis defense. So I'm glad he didn't take that opportunity. So it's nice to see a lot of faces here. I was going to date myself and say that when I was growing up, The Breakfast Club was a movie about people who get sentenced to early morning detention, and it's a form of punishment. And judging from the amount of students here, I can see that it's still early morning punishment. But today I hope that this isn't too bad for you guys. I am still fighting the flu a little bit last week, so bear with me. But today I'm going to talk to you about all the great stuff that we've been doing in our lab over the last couple years. I chose a broad title, Novel Strategies for Treating Cardiac Dysfunction or Treating Myocardial Infarction. And I'm intentionally broad because I wanted to give people a flavor for all the different things that we do in the lab. So today, instead of getting a big long story, let's say like Ulysses, you're going to get the Canterbury Tales, which is just little excerpts from different projects. The big important thing here, though, is that heart failure is the leading cause of death. So this is not anything new. It's the leading cause of death in the U.S., and it's about, excuse me, it's the leading cause of death worldwide, and it's only growing. So you can see here some statistics. This is from the AHA's 2009 website. And you can see here 5.7 million Americans suffer from heart failure. And I'm going to say that it's probably one of the biggest costs to the healthcare burden. So I think they estimated last year it was over $500 billion in healthcare costs for this country alone. It's the leading cause of hospitalization for people over 65 years of age. You can see here about 300,000 people die each year due to heart failure, which is a subset of the 850,000 that die due to cardiovascular disease. So you can see here about each year 500,000 people will develop new or recurrent myocardial infarctions. I would show the heat map, but it would only depress you because it's all in Georgia, Tennessee, Kentucky, the southeast here, due to diet, obesity, genetics, things like that. But we're in the largest concentration of heart disease in the country, probably in the world. So this is really a growing problem. You can see here currently the only cure for heart failure is cardiac transplantation. About 40,000 patients require cardiac transplantation each year. And you can see here only 2,500 donor hearts. And ultimately about one-third of all patients listed for transplantation survive until the heart is available. So what they do now is, and this is the heart failure, this is the work cost. So in missed work and missed labor, it's about $37 billion, but in healthcare costs, it's much more. So this is really, really a growing problem, and it's only getting worse as our population ages and as our diet gets worse. We used to joke when I was in graduate school that the first thing you see when you walk out of Grady is a McDonald's. So a lot of people, they go to Grady for their heart disease, or they have their acute MI, and as soon as they're stable and they're released, they get to go right back out and eat some more fast food. So this problem's only going to get worse as the population ages and our diets get worse. I always like showing this because this is one of the reasons why I started doing cardiac regeneration at the Brigham. I went there, and this is a study, this is a review actually from 2002, but this is a study from the 1970s showing that newt hearts can regenerate. So you know that everybody knows if you grab a salamander, the tail falls off and it'll grow back. And I'm sure you've cut up lots of worms in your biology classes to see the two-headed worms and everything grows back here. But you can see this newt when you make an incision in the heart, what happens is there's a clot forms, then the cells at the border de-differentiate. So they go back into the stem cell fate, they proliferate, and they re-differentiate to give you a brand new heart here. So this happens in newts and a few other species. As I say in all my talks, we're not newts, so our hearts don't regenerate. The heart you're born with is generally the heart you're going to die with. It's kind of depressing, but all those cells in there that are there when you're born, about 80 to 90% of them are there when you're nice and old. So our heart is considered terminally differentiated. Another important thing to understand about cardiac healing is that, well, we call it cardiac dysfunction because it doesn't really heal, but it happens through several stages. And I'll show you the importance of this when we start to design biomaterials and look at our other studies. But, and Jay made this slide, so it made my job a lot easier. But generally what happens is you have this arterial occlusion. So you have a clot formation either somewhere in your body or usually in the coronary arteries. What you get here is a blockage of the coronary artery, and you get this ischemic zone here, this unhappy zone. It's deprived of nutrients and oxygen. Usually it can happen over the course of decades, but what happens usually is there's an acute event where the clot moves and it lodges in a certain area, and you get this blockage, ischemia, so no oxygen, no nutrients. What happens then is the patient gets rushed to the hospital. They run upstairs to the cath lab, and they use a catheter to go in there and give them angioplasty, which clears the vessel. That's great and all, but what happens when you clear the vessel, despite restoring blood flow and nutrients to the tissue, is you get this acute inflammatory response. So oxygen now is the final electron acceptor in the electron transport chain, and you get tons of reactive oxygen species. What happens is your body basically treats the area as a wound. Because there's all this acute damage, so you get this acute inflammatory response, which is macrophages, neutrophils, monocytes all come to the area. And as everybody knows, the job of the inflammatory system isn't necessarily to repair, but is to destroy pathogens. So what happens is these inflammatory cells will go to the left ventricle and start to destroy your tissue. That leads to chronic inflammation. So what happens then is that now that your macrophages have gone there and done all this damage, is that now it's time to repair all this damage. And the way your heart does that is by signaling to the fibroblasts to produce extracellular matrix and divide. So cardiac myocytes, as I said, are terminally differentiated. They don't divide. But you have a lot of lost mass in your heart. You have billions of myocytes that were lost. So the fibroblasts secrete this collagen scar, which gives the heart stability. Unfortunately, it's a non-contractile scar. And then eventually what happens here is that over time, because the collagen does not contract, your heart can't pump enough blood. Eventually the myocytes undergo hypertrophy, which is they grow bigger to try to compensate for the fact that there's fewer of them and they need to do more work, and that eventually will lead to heart failure. So as you can see here, there's specific stages, and there's a temporal signals and cues that happen at each stage, all the way from the occlusion all the way to the onset of heart failure. So it means that when we're designing therapeutics and we're looking at our therapeutic approaches, we have to think about some of these things going forward. So we did this study at Harvard when we were trying to figure out more about cardiac drug delivery and why cardiac drug delivery was so hard. And it turned out that the answer is pretty much staring us and everyone else in the face. These are mice that were injected with a dye that stains endothelial cells green. The red borders are the cardiomyocytes, so these black areas are actually the cells, and blue is nuclei. And then we sacrificed the animal, fixed the tissue, and sectioned it out. And as you can see here, the heart is really vascular. I'm sure you didn't really need a picture to show you that, but it helps when you're doing some modeling, is that the heart, each myocyte is completely surrounded by a network of capillaries. You can see about 5 or 6 capillaries each. What you can't see is that this is 3D, and there's even more interaction as you move down. So what does this mean here? It means that it makes retention of things that are very small extremely difficult. And there was a paper here that we did when we were at Harvard. This was done actually with in collaboration with the Laufenberger lab. All of you who are reading modeling papers of EGF and all those wonderful things can see all the work that the Laufenberger lab has done at MIT. But in short, what happens is they modeled what happens when a cell in the heart produces a local growth factor. In this case, it was heparin-binding EGF. because it is the Laufenberger lab. And as you can see here, this green cell is the cell that's producing the factor, which is a cardiomyocyte, and these red areas here are the capillaries. What this picture shows, and this picture here also confirms, is that the capillaries act as a huge convective force. Is that anything, the highest concentration of any drug that you're going to find is going to be in the capillaries, even if the cell itself is producing it. So that means that anything you deliver to the heart will likely be carried away in a matter of seconds or minutes. So that makes it very difficult when you have a long process like myocardial infarction, which can take days or weeks to manifest itself in the injury. So you can see here there's really a strong need for retention of local therapeutics in the heart. And when I came to Emory, this is one of the things that I wanted to focus on. And wouldn't you know it that I happen to be a fan of Naren Murthy from some of the work that he'd done with Pat Staten and over there at UW. We had done some similar things here, and just so happened that he was working on a polymer which I thought might be able to do this for us. So what we had been using in the past is hydrogels, which are hydrophilic. They're great for large proteins, but for small hydrophobic molecules, they don't do you much good. So I teamed up with Naren, who had recently developed Polyketal particles, which were so named here for this pH-sensitive ketal linkage. So traditional molecules, I'm not going to show you this, but traditional molecules such as PLGA, they degrade into lactic acid and other acidic byproducts. In fact, it's lactic acid, PLLA, and things like that. So they degrade into glycolic and lactic acids, which can cause tissue inflammation, which is probably not a good thing when you're dealing with already a nice level of tissue inflammation. So these are neutral. You can see here this ketal linkage, which is acid sensitive. It doesn't really degrade in water, except when it's exposed to acidic conditions, you get breakdown into a diol and acetone. The main one that we use is called PCADK, which degrades into cyclohethane dimethanol, which is the diol, which is an FDA generally regarded as safe product as a food additive, and acetone, which is also on the FDA approved list. You make it naturally and you breathe it out. So this polymer here degrades under acidic conditions into neutral byproducts, which is just great. And you can see it's summarized here. And also, the hydrolysis is tunable. I'm not going to show any of this data, but Stephen Yang and Naren did a great paper showing that you can change this R group. Whoops. You can change this R group up here by changing the hydrophobicity of the molecule. You can totally make it for a half-life of days, weeks, months, and I think they had one that would degrade even in a year. So here's some SCMs of them here. You can see we make them microparticles, and I'll explain why in a minute. But you can see here when you create this emulsion, you get these really nice spherical particles. Here the mean diameter is probably about 2 to 5 microns. So the initial study we did was we chose this P38 inhibitor, and I'm not going to, like I said, this is the Canterbury tails. Maybe this will be the Miller's tail or something like that. But we're going to keep it really simple. I don't want to show you the 8,000 pieces of data that went into this. But we use this small molecule inhibitor here, too. It's SB, one of these SB compounds. It's a P38 inhibitor. So P38 is a kinase that's involved in inflammation. It's implicated in cell death, the inflammatory response. And in fact, mice that have a dominant negative P38 alpha survive better after ischemia reperfusion. And there was a lot of papers showing that if you gave mice this inhibitor orally over the course of a couple of months, that they actually improved function following MI. Unfortunately, in this paper, they pretty much washed these mice with the inhibitor twice a day, every day for about 3 months, which in a human would basically mean that you'd probably be walking around and drinking it constantly all day, which really isn't feasible. So we thought, wouldn't it be great if we could figure out a way where you can actually get the same effect just doing it once? So we took PCADK, which is shown here, and here's the breakdown products. And you can see combined it with our small molecule inhibitor in a single emulsion. Because the inhibitor itself is hydrophobic and the polymer is hydrophobic, we were able to put them in a single emulsion over some PVA. You can see here we homogenize at a very slow speed. So we got these kind of big particles, and we always joke that this isn't really nanomedicine because it's micron particles, so we call it micromedicine. But sometimes nanomedicine isn't really good. So you can see here we did a permanent occlusion of the left descending coronary artery. So we took, we verified all these wonderful things in cell culture. They went into macrophages. The macrophages would release it. So what happens here, I didn't mention, is that these particles will be internalized by inflammatory cells, and once internalized into the phagosomes with low pH, the particles will hydrolyze and it will disrupt the phagosome and release into the cell. So you get this intracellular delivery. So these were delivered after permanent occlusion of the LAD. So basically what we did is we just opened up the chest of a rat, tied off the coronary artery to simulate a heart attack, and then injected these into the border zone, trying to simulate what we think might happen in a clinical situation. So I'm not going to get into all the nitty-gritty details, but what happened was that we saw after 3 and 7 days that our inhibitor significantly decreased p38 phosphorylation in the infarct zone compared with the free inhibitor or the empty particles. And that got us thinking, well, we just wanted to make sure that these particles are still really there. So what we did is we loaded these particles with rubrene, which is a nice fluorescent dye. It's hydrophobic. And we injected these into the heart after ischemia perfusion. And then we harvested these at 3, 7, and 10 days and imaged it. And you can see here, this is a 3D reconstruction of a section. So these were section slides that were stained green, which is for troponin, which will stay in the sarcomeres of the cardiac myocytes. And this yellow, big giant yellow thing here is a loaded particle. And this is several days after injection. So remember I said we make them microparticles. And we make the microparticles because we want them to stick around for a long time. So you can see here this is 3 and 7 days, and here you can see a little bit better. There's a bunch of little particles that are sitting around in the heart. So we're pretty sure that when we inject these particles in the heart, they're actually staying in the heart, which is big if you want to create a drug reservoir in the myocardium. I'm not going to show the data, but we looked at 10 days and we still saw them there after 10 days. So we're pretty sure that not only are we delivering it to the area, but it's sticking around and it's releasing the drug over time. So to cut to the chase, and we'll have a happy ending here, we looked at function. So we looked at lots of other markers like inflammation and decreased inflammation. But ultimately, all that really matters is what happens to the function of the heart. So this is a fractional shortening, which is a measurement of how well the heart is contracting. These were done by MRI and echo. And you can see here, this is 2 bars. The white bars here are day 7, and the gray bars are day 21. You can see here on the y-axis is the percent of fractional shortening. So you can see here, these sham animals, they contract about 50 to 55%, which is what they're supposed to do by measuring peak systole and peak diastole areas. You can see here when you have an MI alone, there's a significant decrease both at 7 and 21 days. There was no effect of our P38, this MI plus P38I, which is the free inhibitor, and the MI plus PK, which is our empty particles. There was no effect. Now, interestingly, when we looked at our P38 loaded particles, this PK, P38 sub I, you can see here at 7 days there was actually no functional improvement, which got us a little worried and started to panic a little bit that despite all these positive results, nothing was happening. However, when we looked at 21 days, you can see here there was a significant improvement, and this was also significant against the MI alone. So you can see here over time, these animals improved their function. And for the reviewers, remember, for those of you that are in my class, I talked about all the wonderful things that reviewers always make you add. They wanted us to show that we could beat the industry standard, which was this PLGA. So we created sized match and loading match PLGA particles. And I'm not going to show you all the preliminary data from that, but basically they themselves induced P38 activation in a lot of cells. And you can see here there was no significant improvement in function. So the take-home message from this slide here is that although there was no acute improvement at 7 days, at 21 days there was a significant improvement in function. So we went back and we looked at all of our data, and we realized that by inhibiting TNF and superoxide and hydrogen peroxide and all these things, that perhaps we were having an effect on remodeling. I don't think this actually worked. I tried to load some movies in here because a movie's always worth 1,000 words, but it didn't seem to want to work today. But this is how we get our images here, is that we look at MRI slices from systole and diastole, and if I had some magic now, you'd be seeing these contract, and we can measure the areas at each one. But we were looking for a reason why these animals got better. And when we sectioned the tissue, we looked at collagen staining. So, I showed you that initial slide that had longer-term things being remodeling and hypertrophy and things like that. And you can see here, remember when I told you guys that the heart basically turned into a collagen scar. You can see here, here's the left ventricle of a regular MI mouse. Or rat. This is 3 weeks later. The red is collagen. The yellowish is these myocytes that are still alive. And you can see here, about most of the wall turns into this collagen scar, which is non-contractile. We actually measured a lot more than this. This is just one area to show you, but we measured this in lots of sections, and it's about 30 to 40% of the heart, of the left ventricle, is a scar. As you can see here, percent fibrosis. If we have our free inhibitor, there was no effect. If we have our loaded inhibitor, our empty particle, which is just PK, there was no effect. But you can see here our loaded inhibitor shown here significantly inhibited fibrosis, which we think allows for better contraction. So we didn't have an acute effect, but we had this chronic effect, which was great, but it made us want to think maybe we could have done better on the early time point as well. And I'm going to switch gears really quick and talk about this early time point, because what's good for the goose is not good for the gander. I showed you the specific temporal constraints here, and you can see here at later time points we have this fibrosis, but at early time points what you have is cardiomyocyte death. Initially about 0 to 72 hours you have a huge amount of cardiomyocyte death, which might be what's limiting the function at early time points, not fibrosis. So our lab is also interested in reactive oxygen species. Emory has a wonderful core for measuring reactive oxygen species, and it's one of the top places in the world for reactive oxygen species. As a way of a little bit of brief background, low oxidative stress here stimulates growth. You need it to survive. But high oxidative stress stimulates apoptosis and necrosis, in addition to a whole host of other negative things. But reactive oxygen species, specifically superoxide, has been implicated in cardiomyocyte apoptosis in particular. And you can see here, low levels stimulate growth. We have transient adaptations at these moderate levels, but at these huge levels, you just get apoptosis and necrosis. Interestingly, superoxide levels increase in the infarct zone right after MI, and the RNA levels of the endogenous antioxidants, such as SOD, sharply decrease after MI. And there have been some controversies in literature referring to SOD therapy. So SOD overexpression protects the heart from ischemia reperfusion injury. But antioxidant therapies with the actual protein, SOD and Catalase, which takes hydrogen peroxide away, they've been attempted at different laboratories, but the results vary widely. Some of them say it works great, others say they can never get it to work. And one of the big issues is unfavorable pharmacokinetics. So the half-life of SOD protein is about 2 hours. So basically, even if you can get it to stay in there, it's rapidly inactivated by its own product, which is hydrogen peroxide. And it also leads to instability of the protein. So you get really poor delivery kinetics. Also, improper delivery. SOD is a very large active protein, so you can't really get proteins inside of cells, which is probably going to hurt you a little bit. Lots of laboratories have tried to address these issues by attempting different delivery methods. You can see here PEG coupling, liposomal encapsulation, lecithinizing them, even coupling of heparin binding domains. I've actually tried that with other proteins to get it to stick in the area. But really there hasn't been a lot of work on microencapsulation of SOD. So we went into this wondering if we can improve the delivery of this endogenous antioxidant protein by encapsulating it within these Polyketal particles. So once again, we use our friend here, PCADK. This is a little bit different though because it's a protein. We did a double emulsion. The first emulsion is done in a small volume of PVA here, and we've done it different ways. But basically the first emulsion is to protect the protein itself. So you create these small little particles either by sonication or by fast homogenization. And then you put that into a second emulsion containing PVA to get your actual particles. So you get these little tiny particles, and you get particles within particles. And then the same delivery concepts we're working on here, which is hopefully phagocytosis and intracellular delivery. And you can see here we did a little bit different this time. We used ischemia reperfusion, which is when you tie off the LAD for about 30 minutes. LAD is the left anterior descending coronary artery, and then you release it. So that actually, we believe, simulates the clinical situation even better. It's a very common technique that's used, but that's when you get that huge influx of oxidative stress. Direct injection of the microparticles, because it's an open chest procedure, we can put the microparticles directly into the heart. And in this study, GoCool did 3 injections into the border zones. So you can see here, the infarct core would be the middle. The border zones are obviously around the borders, which is where reperfusion is thought to best occur. And then this little schematic here again shows pretty much what we're hoping is that these particles will sit inside the tissue between these myocytes, either get engaged by macrophages or release the product by itself. And you can see here, we did a lot of function. This study, instead of just measuring function at 7 and 21 days, we measured baseline function. So we could see how the animals were before the surgery. And then we measured echocardiography at each time point, and MRI at certain time points to look at function over time. And we also did biochemical data like oxidative stress and cytokine responses, things like that. In vitro, we looked at macrophages, and you can see here we stimulated macrophages with PMA just to make sure our SOD in this double emulsion procedure was still active. So you can see here, this is measuring extracellular superoxide, and this is with HPLC, so it's pretty quantitative. And we stimulate our macrophages with PMA. You get about a 1.5-fold increase in superoxide production, which is blocked by extracellular SOD. This is just the free protein. There's nothing special about it. But it's what you'd expect. It's a good control just to show that you can block with SOD. If you look at our empty particles, you can see here there's no response. These 2 blue bars here, there's no effect of empty particles on PMA-induced superoxide stimulation. You can see here this middle bar here. But if you look at our PKSOD, which is our Polyketal encapsulated SOD, at 2 very low doses, this is 0.25 and 0.125 milligrams per mil, so we're talking tiny, tiny doses here, you could see a complete inhibition of extracellular superoxide. That is not really cool. That's just about what we expected, because superoxide can probably freely diffuse in and out anyway, and this is outside this cell. The really interesting thing is when we looked inside the cell, you can see here there's much more of a response inside the cell. There's about a 4-fold increase in superoxide production inside the cell, which is not blocked by the free protein. So proteins don't cross into cells, so we didn't really see much inhibition. If you look at our empty particles, once again, there was no effect of these empty polyketals on scavenging the superoxide. But interestingly, when you looked at our PKSOD particles, you can see here, even at this 0.25 dose, and even a little bit at the 0.125 dose, there was a significant scavenging of superoxide, or a significant inhibition of superoxide. So what this means is that the macrophages were able to phagocytose or engulf the particles, and that the active protein was released inside the cell. So not only do we think we have a way to get sustained delivery of our protein, but we think we also have a way to get it inside of cells. So I'll skip all the exciting part. I'll just show you that in vivo, when you look after ischemia reperfusion, this is a dye that measures oxidative stress. This is bad advertising for Narain, because we didn't use his reactive oxygen species dye, but this is just regular dihydroerithidium, which will react with superoxide to form ethidium bromide, which anybody who's worked with DNA knows that it will bind to DNA and fluoresce. So you can see here, after ischemia perfusion, this is heart tissue. There's a lot of superoxide being formed. If you just give free SOD to the animal, nothing really happens, but you can see here our PKSOD completely prevents superoxide formation in the myocardium. And this led to acute changes. So this graph is a little bit different. On the y-axis is actually a change in function rather than the actual function itself. We measured baseline and then measured at 3 and 21 days later, and you can see here, this is at 3 days, ischemia reperfusion caused about an 8 to 10% decrease in function. And that's percentage points, I should mention, not percent. So that means if the animal had 50% contraction on day 0, it now had 40% contraction on day 3. And you can see here our PKSOD significantly improved that. Unfortunately, at 21 days, what we saw is our PKSOD did not really have much of a significant effect anymore. But when we combined our PKSOD with our P38 particles, so if we attack both ends of it with apoptosis and fibrosis, we actually get a significant response. So this would suggest that A, there's not one magic bullet that's going to really do it, and B, we really need to think more about spatial and temporal delivery, because there are specific time courses to these things. So the PKSOD, while it worked great early, it didn't work great very late. And the opposite was true for the P38 inhibitor. It worked great chronically, but didn't really have much of an acute effect. So this got us to thinking, is there a way we can try to do both? So this is brief data. I'll just get into this really quickly, but this is data from Indu in the lab looking at cutting off the superoxide at the heart. So rather than just giving a protein which may not last a long time, is looking at ways to knock down the enzymes themselves that make the bad things. So this is a Polyketal here with siRNA particles in it, and we can complex RNA with this molecule called DOTAP, which is just positively charged. It's called ion pairing. It's just to get this into the particle, because a negatively charged piece of RNA won't really get into particles well, and it also will be subject to degradation. So we pair it up here. We can put it inside polymers, and you can see here, this is a little schematic from Narain's lab, where you can have your Polyketal particle, the siRNA, gets introduced to the cell will phagocytose it, the RNA will be released, and it can go to the nucleus. So we looked at, you can see here, siRNA therapeutics have a lot of promise because you can specifically target one protein, as opposed to small molecule inhibitors, which tend to be a bit ubiquitous. But delivery problems have really not advanced the field very much. And you can see here, we looked at NOX2. So this NOX2 is a subunit of the enzyme NOX, which makes superoxide. It's called NADPH oxidase. It's one of the enzymes that is actually responsible for making the superoxide. So rather than scavenge the superoxide after it's formed, we try to cut it off at the pass. And you can see here, this is just really preliminary data, but here's empty particles, and this is our scrambled siRNA particles. And you can see here, this is normalized NOX2 levels. And you can see here, this is after 24 hours. You can see about a 40 to 50% decrease in NOX2 expression. So we're hoping that systemic delivery of NOX2 siRNA to the MI, or perhaps local delivery with the methods I've shown you in the past few slides, that we can actually stop the source of the radicals before they even come out. So now I'm going to switch gears a little bit again and talk about more exciting stuff, which is affinity delivery. And I might go through this a little bit quicker because there's a lot of background and it's already been published. But one of the things that we saw from our previous slide here is that there's two molecules which improve the function, one early and one late. And we actually had to create two microparticles to do this. So we thought, wouldn't it be great if you can do it on a single microparticle, to have one microparticle that can release two things with different time courses. And it just so happens that Jay was digging through the literature and found this nickel affinity paper here. So nickel affinity columns are used to purify proteins. What's normally done is you express your protein in a cell with a histidine tag, and then to purify your protein from all the proteins that the bacteria or yeast are making is you run it over this nickel column. Nickel NTA column, and it will bind to histidine tagged proteins right here. And we thought, wouldn't it be great if we could apply this same theory to a microparticle? And as it just so happened, there was this molecule here called DOGS NTA. If you want to know what DOGS stands for, you can look it up. It's a really long fatty acid chain, and it's got a very long name I can't even begin to get into. But basically what it is this long fatty acid hydrophobic chain. Then you've got this hydrophilic head group here, which is NTA, nitrotriacetic acid. And what happens here is that what we hypothesized is that because this is hydrophobic and this is hydrophilic, if we put these into our emulsion, the hydrophobic part would partition into the microparticle, and the hydrophilic part would stick outside of the particle into the aqueous phase. And it just so happened here that, I won't get into too much detail, but you can see here's a schematic. There's our microparticle, and here's our nickel NTA. This is their NTA. You combine it with nickel. And we're hoping that we combine his-tag proteins to the outside. So we can get dual delivery. We can get delivery of something from the core that's hydrophobic and something from the outside that's hydrophilic. And without getting too much into it, we made these particles, incubated them with nickel chloride to get nickel onto the NTA, and then once these particles are made, we can freeze dry them and we can actually put them into an aqueous solution containing the protein. So that way your protein never has to see any organic solvents or anything like that. And even the great thing about this is when you're done, you can spin it down and recover the unbound protein. But you can see here, Jay incubated given amounts of protein on the outside and ran a quantitative ELISA to figure out the efficiency of capture. You can see here nanograms of GFP loaded on the x-axis, nanograms of GFP captured on the y-axis. And the take-home message here is that this is about 40%. So we get about 40% of our protein bound to the outside. And I should mention in our initial SOD studies, we were happy if we got 5% of the protein inside the microparticle. Because it's double emulsion, the protein can seep out into the aqueous phase of your emulsion. Here, because the entire thing is done in an aqueous solution, we get about 40% binding. And there is some saturation. It's dependent too on the amount of NTA. You can see here if you have 1% NTA versus 10% NTA, the binding changes. But the end result is you can get about 40 to 50% of your protein bound to the outside of the particle. And what does this allow for? Well, you can see here release from the outside. This is release study using GFP and our SB compound from the inside data. You can see here our SB compound has a half-life of about 7 days. That means about half of the inhibitor is released in about a week. And it's probably mostly either diffusion or particle hydrolysis. Whereas here, you can see the GFP, his-tag GFP on the outside of the particle will release with a half-life of about 2 hours. And this was actually done in serum. So probably ends up pushing a little bit. If we did it in PBS, it might be a little bit different. But what this shows us is that we can encapsulate two completely different proteins, one hydrophobic, one hydrophilic, and get absolutely different release profiles, which is exciting for us because then we can maybe go back and look at some of our original studies to see if we can get the same results from this one particle instead of from two separate particles. Also, what this allows us to do is we can incubate a variety of different his-tag proteins on the outside of the polymer. We don't have to use just one protein. And in fact, we're doing some collaborations now with Dr. Young-Sup Yoon in Emory Cardiology to see if we can generate IPS cells, which is induced pluripotent stem cells, with a cocktail of factors on the outside. The other thing that this can be used for is it can be used for targeting. I won't get too much into this because this is probably just a small sampling and we didn't even choose the best protein. But instead of using antibodies, you can use targeting proteins. If anybody who's taken the IBS class knows that cadherins are on the outside of endothelial cells, mainly these VE cadherins, vascular endothelial cadherin, and they're used for cell-cell communication. They bind to cadherins in other cells, and they're transmembrane proteins, so when the cadherins bind, there's some calcium-dependent processes. The curious thing is that they form homodimers with other VE cadherin, so we hypothesized that this might be a great way to try to target these microparticles to endothelial cells. So we found a his-tagged VE cadherin fusion protein, and we put it on the outside of our particle. It incubated them with endothelial cells. And you can see here Jay did some confocal. Phalloidin just shows you the cell. The red is the particle, the blue is the nucleus, and these are endothelial cells. These are HUVECs. And you can see here little red particles associated with the cells. And we did all sorts of different cells. We did HUVECs with VE cadherin particles. You can see about 40 to 50%, as opposed to just HUVECs. This is just nickel NTA particles. There's no VE cadherin. And also we did cardiomyocytes, both with and without cadherin. Cardiomyocytes don't express VE cadherin. So you can see we get about a 2.5-fold increase in endothelial cell targeting. So this is exciting because now we might be able to use these particles for things like hypertension and atherosclerosis, which mainly involve endothelial cells. Finally, in terms of targeting, this is work that's being done by Warren Graydon Lab. There was a paper a few years ago that showed you can use sugars to target cardiomyocytes. And Warren has now spent the good part of a year trying to figure out how to get this sugar, which is N-acetylglucosamine, conjugated to a microparticle. And you can see here we've employed the same technique, which is conjugating the sugar to a big hydrophobic molecule, so we can create this partition again. And then hopefully we now have these particles made and labeled, hopefully that they'll bind to lectins on the outside of the cardiomyocytes and be internalized to cardiomyocytes. And this would be a huge leap in the field because cardiomyocytes aren't phagocytic cells. They don't eat particles. They don't do lots of things. They just contract. My neuro friends always used to like to tell me that all the heart does is pump. And it's true. All these cardiomyocytes do is contract. So if we can figure out a way to get proteins to the cardiomyocytes, we can in fact give them either positive contractile factors or positive survival factors directly after MI. So that's the end of this tale here. The polyketals make excellent delivery vehicles for treating MI. We can get release of multiple factors simultaneously. We can now get temporal control. We can get spatial control here for targeting different cell types. We can deliver anything from proteins, small molecules. Now we have siRNA, and we can sustain the delivery for several days and weeks. So we're still doing some polyketal work, but we've now gone on to new biomaterials because these are invasive delivery techniques. So I should mention that intramyocardial injection, while it's being done in humans, still requires a certain degree of technique. You need a trained physician who knows how to use a catheter to go in there, and actually you have to put the catheter in through the artery and snake it all the way down into the left ventricle, which is a tricky procedure sometimes. And then you have to know where you're injecting it because you don't want to inject it randomly in the left ventricle. You want to inject it in the injured area. So this got us this thinking, wouldn't it be great if we could figure out a way to target these directly to the infarct itself? And I wish I could take all the credit for this idea, and maybe if Narain weren't here, I probably would. But some of this came through with Narain Murthy, who was doing this with cancer therapeutics called autocatalytic targeting. And we sat down one day and thought this might be a great idea for infarct delivery, and I'll get into this in a second. So I mentioned to you earlier that cell death following MI is a big problem. You can see here all sorts of, there's this infarct zone here, there's the peri-infarct, there's the border zone, but what happens here around the border zones and the infarct is you get necrosis, apoptosis, you get autophagy, which is this totally different creature. But in the remote myocardium, there's almost no cell death acutely. There's chronic, there's delayed cell death, after days and weeks because of adaptive mechanisms, but acutely, there's no cell death. And you can see here's a picture of a human heart with that's had an MI, and you can see a lot of cell death. And here's a little picture, and Jay dug this one up, which is good, because it shows the two processes of apoptosis and necrosis. I'm not going to call on anybody like this for class and ask you which one is necrosis, but clearly the one on the outside here is necrosis because it's not controlled. It's releasing everything into the extracellular environment, whereas you can see here apoptosis, everything is being packed into vesicles and processed. What this means is that after MI, you get this huge increase in necrosis, that there's a lot of things being released into the extracellular environment that shouldn't normally be there. And one of those things is DNA. So Narain's autocatalytic targeting molecule targets DNA, and it's for killing cancer cells. But we thought we could probably use this to try to target DNA in the infarct zone, because in healthy tissue, DNA shouldn't be there. And I like this because Jay made it. We're hoping that you can inject it intravenously, and it would all accumulate just in the heart. And you can see here another little schematic here. It would accumulate over time just in the infarct zone of the heart. So infarct-specific targeting. And like I said, these are tails, so I'm just going to show you we use a hoaxed molecule. Hoaxed is a DNA binding agent. It's been used in clinical trials for measuring angiogenesis and even for cancer, anti-cancer treatments, but it binds to the minor groove of DNA with relatively high affinity. And the great thing is it can be modified. So what they did here, you can see here's the hoax. What they did is they added a polyethylene glycol linker. So this was anywhere from 4 to 11 peg chains. What this did is it made it cell impermeable. So hoaxed is cell permeable, but adding these peg chains made it completely cell impermeable. So it doesn't bind intracellular DNA. And then you've got this reactive handle here. The initial studies, we use this IR786 dye, which is an infrared dye, which we can use an in vivo imager. You see here low tissue attenuation during imaging. So our theory here is that there won't be any extracellular DNA unless there's an injury in the tissue, and that this doesn't bind across the cell membrane and bind intracellular DNA. So I won't get into the whole story, but there's a lot of pictures, and it doesn't bind to cells, so we incubate it with cells and we don't see any nuclear fluorescence until you permeabilize the cells. So we went ahead and we did this in vivo. So you can see here, once again, we have this coronary artery ligation. And this time we close the chest and remove the ligation, reperfuse the heart, close the chest. Just like you would see in the clinic, the patient gets reperfusion, happy, sent on his way. 2 hours later, or 2 to 24 hours later, Jay and Milton injected the hoaxed IR786 conjugate intravenously, and then after an hour sacrificed the animals, perfused the organs, and fixed it for imaging. And there's really a lot of pictures, but I'll only show you the ones that matter. There wasn't really much staining in the lungs. There was a little bit in the liver, as you would expect, but it was not specific staining. It was just sort of some diffuse staining in the liver, and there was no staining in the kidneys either. So we're pretty certain it gets to where we want it to get to. When we image the myocardium, you can see here, this is a sham-operated animal that received IR786, and here's an MI animal that got the IR786. And this is pretty, it actually turned out a little bit better than we'd even hoped. I took out the one where the suture is, but basically the suture is probably right about here. So most of the damage is below that in the LV. And you can see here we get a pretty strong signal just in the injured area. If you quantify that, it's about a 2-fold increase at 2 hours. There wasn't really much change at 24 hours, but I must say that it's still not all that quantitative because it's just looking at images. And we're actually going to get some real proteins and do some real quantitation. But the exciting thing is that when we looked at these under planimetry, so when we dissected the tissue and we sliced them to see exactly where we were in the LV, you can see here, here's the RV over here. There's no staining. Here's what they call the remote myocardium of the LV. There's no damage there. It's all concentrated along this infarcted area of the LV, just like that picture I showed. And if you took an infarct image of a real rat or human, that's probably what you would see for the dead tissue. So we're pretty certain this is binding to the dead tissue with a fairly high affinity, and there's almost no binding in the remote tissue or other tissues. So we're pretty excited about this because if we figure there's a way that you can treat patients even before they get to the hospital, maybe on their way to the hospital with some kind of anti-inflammatory agent. Right now they tell you to pop an aspirin or something like that, but really until you get to the ER, there's really nothing else that you can do. So we thought it might be great if you could have something that you could inject intravenously, which would attenuate cell death or at least take away some of the inflammation. So like I said, it's allowed for targeting of the damaged area via an IV injection. There's still a lot of work that needs to be done. We can conjugate proteins, particles, maybe even cells to this using our lectin approach or something like that. But obviously there's still a lot that needs to be done, like determining the kinetics, how long it's there, and if we can actually use it to deliver anything meaningful. And right now we're in collaboration with Andres Garcia to deliver VEGF to see if we can promote angiogenesis in the infarct zone. So I'll end the talk here with a few more slides talking about how we find new therapeutics. And I really want to touch on everybody else that did work in the lab here. So this is more about finding new therapeutics, or finding more about existing therapeutics. So that I show, you can search the literature and find cool drugs, and it's fun to inject biomaterials into the heart and play with them and find new materials and new things to do. But ultimately, we need to know about these things such as cell specificity, timing, and function. And we don't just guess it. There's actually studies that need to be done to figure out what cells need to be targeted and when. I told you in the very beginning, we started by saying that myocardial infarction is a very progressive disease, and it takes weeks and days and weeks, and lots of different cell types are involved. So everybody, when I go give talks to places, they say, well, how do we actually figure out new therapeutics? And if you're got lots of money, like Eric Olson at UT Southwestern, you can make a mouse every week that has a new microRNA or a new protein, but we don't have that kind of time, energy, or money. So in the lab here, we use special mouse models and cell culture, and to get back to the basics. So just so people don't think we only do materials in the lab, we try to figure out interesting new things about cells, specifically stem cells, and to see if we can develop new therapeutics. So I'll say we have transgenic strategies, and we use this strategy to look at timing and cell specificity. I won't get into the data here, but we've created mice that have this alpha-myosin heavy chain promoter, which is cardiac specific, and then this MER-CRE-MER, which stands for mutant estrogen receptor. So it binds to something called tamoxifen to turn on this protein CRE. So your body doesn't have a lot of tamoxifen. You can imagine a male mouse doesn't really have a lot of mutant estrogen receptors. And what you can do then is you can take CRE, this LOX-CRE technology, and you can use it to change the genome of an animal. So what we did in one study is we have this GFP and a stop codon, and then the human catalase gene. So I didn't talk too much today about catalase, but catalase is another antioxidant that takes away hydrogen peroxide. And you can see here when you inject tamoxifen, you will get CRE, and it will remove this stop codon and allow you to get catalase expression. And this will be just in myocytes. And using this approach, we've been able to look at the timing of delivery, so we could say when should we deliver catalase. You can see here 0 days, -7 days, we can do 14 days, and this paper was just accepted. But what basically happens here is if you do it acutely, so this is animal that got catalase just before surgery, and here's one that got it a week later. You can see here there's almost no change depending on when you give it acutely or a week later, which suggests that maybe catalase isn't the best protein to give acutely after MI, which you wouldn't know by digging into the literature because there really has been a lot of work trying to figure out why catalase doesn't work early. And it's because we found that it doesn't really matter when you do it. It doesn't change cell death. It actually changes fibrosis over time. I won't get too much into that, but you can see here there's absolutely no difference in delivering the timing. We now have macrophages that overexpress catalase as well, so we can look at cell specificity. And finally, I'll end it with some stem cells. All these different kinds of stem cells, I'm sure you've all heard of, embryonic, skeletal myoblasts, they all have benefits and they all have downsides. For the function of MI, we've really only looked at endothelial progenitors, mesenchymal stem cells, and cardiac progenitors. These are tissue-specific adult stem cells. And these three are really the only ones that have had clinical trials done on them. And the clinical trials show an acute benefit, but there's really no long-term benefit. And moreover, they really have no idea why it works. They just know that you inject cells and magically the heart happens to heal, which is good for those patients for 3 months, but doesn't really help them long-term. Because you can see here there's paracrine effects, there's making new myocytes, there's new vascularization, there's repairing the cells. So these are these tissue-specific cells here. You can see these little tiny cells. This is an adult heart. These are big adult myocytes. And these are these little tiny stem cells that sit in the heart. Some of our collaborators have identified them, and we actually use magnetic beads to pull them out of the heart, and we can study them in culture. And one of my students, Go Cool, looks at what happens when you expose them to superoxide. And it turns out, unlike adult cardiomyocytes, when you expose these cells to superoxide, they respond by acutely upregulating their antioxidants, SOD1, SOD2, glutathione, and peroxidase. And they do this probably what he thinks is by activation of AKT. So by studying the pathways by which this protective effect occurs, we can use it to try to direct delivery of these proteins or delivery of these factors in vivo. Or we can modulate stem cell therapy itself. We can deliver stem cells that have AKT activated in them. I won't get into this, but oxidative stress regulates angiotensin receptors, which is also really bad because angiotensin 2 is critical for inflammation and hypertension. When you expose these cells to hydrogen peroxide, their Ang2 receptors go way up, which would end up negatively modulating these cells and preventing their differentiation and preventing their survival. So if you're going to inject these cells into an area like the heart, which is rich in superoxide and oxidative stress, you really have to understand what's going on. Finally, if we look at mesenchymal stem cells, the complete opposite happens. When you treat mesenchymal stem cells with hydrogen peroxide and oxidative stress, it actually drives their differentiation. These are mesenchymal stem cells which have been treated for a week with hydrogen peroxide, and we measured some endothelial markers. And you can see here that redox or oxidative stress seems to drive endothelial differentiation of these cells. So why am I showing you this? Well, cell therapy is cell-specific. At Emory, we have this thing called preventative care and personalized medicine. And I think this is really going to be one of the next forefronts in medical research is personalized medicine. Is when a patient comes in there and you do autologous cell therapy, it means when you take their cells out, culture them and re-inject them back into it, you really have to think about what kind of cells you're using them, when you're injecting them in there, and what they're going to be exposed to, because you need to understand how these cells respond to their environment before designing therapeutics. And you can see here we found that progenitor cells acutely activate protective mechanisms, but oxidative stress may drive differentiation of mesenchymal stem cells. So now we're going to go back and we're going to see if we can modulate these pathways in vivo with materials to see if we can drive endogenous and exogenous repair. So with that, I think I've bored you enough this morning, but I want to thank all the people that did the work. I have a lot of great people in the lab. Milton Brown does all my animal surgeries for the last 4 years, and probably about 1,000 animals, something like that. Jay and Gokul are fifth-year students about to go on to bigger and better things. Indu, Archana, Warren, Carl, all in the lab. We love lots of new people too. Also the free radical, the MRI people that helped us out. And finally my collaborators here at Georgia Tech, Narayan Murthy, specifically in his lab, Stephen Yang and Scott Wilson. Andres Garcia, who helped with the VEGF study that didn't show the his-tag VEGF delivery, but that's what we used for our dual delivery. And we have more collaboration with him. And Melissa Kemp, who helped us measure inflammation in response to our particles. So, and our funding sources. Thank you.
We have time for a couple of questions.
Don't be afraid, you can ask anything. Maybe not. We'll see.
Okay, nice work, Mike. I've got one question for you. When you're talking about the nickel tag, or the his tag nickel affinity protein system, so that releases in a matter of a couple of hours. What if a couple of hours is not ideal for protein release or for delivery to that system?
That is something we've thought about. We're, we're trying to figure out ways to slow the release, maybe by, I guess using different. You can maybe use different metal chelators, or, that's also his tag GFP. So maybe larger proteins might not diffuse away as quickly, but that is an issue. We're trying to figure it out. We haven't quite got the solution to that, and I'm sure Jay is going to stick around for another 6 months to figure out how to get even more levels of release. But that was also under serum conditions. So I think if you do imidazole, or if there's his tag proteins in the microenvironment, those can also change your kinetics in vivo. So.
Thanks for a great talk. I was curious about your results with the mesenchymal stem cells.
Yes.
Have you seen them differentiate down any other pathways in response to stress? Or
We've looked at smooth muscle, and we actually see smooth muscle markers going down. We looked at cardiac markers, and they don't change, and fibroblast markers go down. So it seems to be pretty specific for driving endothelial, but we did not look at bone or other markers. Eventually we're going to have to look at that, because that's going to be the primary issue, is if we're driving bone growth.
All right, can we thank Mike for a great presentation?